Good morning to everyone. It's Thursday morning, 10 a.m. Central Time, so means enjoy science. Thank you all for being here. Enjoy science. As you know, it's already 18 months old, so we start having a history. And of course, I have all the time to thanks to the Chief Scientific Officer, Dr. Giulio Draeta, for hosting this, to my friend Alessandra Ferraioli. I co-host with her Enjoy Science, and certainly to Liz Kent and now Emily Lang. Emily is a new addition in the team, and thank you both for the wonderful job, because you are keeping Enjoy Science running so smoothly. I want also to thanks to the people who attend. As you know, there are already thousands and thousands of attendees. I can tell you cumulative number past already 10,000 for Enjoy Science. And I want to say, without you who are attending and choosing this webinar, it cannot happen, Enjoy Science. This is for all the family of MD Anderson, from inside the institution, from outside the institution, from United States and from other countries. And in fact, we all are very happy because there are wonderful speakers who tell us about the wonderful science, clinical and basic, from MD Anderson. Today, I have the pleasure to welcome a colleague and a friend, Dr. Anirban Maitra, and certainly everybody here and outside abroad knows Dr. Maitra, knows Anirban. Why? Because he's a wonderful scientist, he's a wonderful colleague, and he's a wonderful collaborator. So he, I don't know if you know, he finished a medical school in India, and then he attended excellent background and excellent training in the United States, Southwestern, Johns Hopkins, and we had the good luck to have him here for, I think, already 10 years, how time is passing. And Anirban, it's a driving force behind the science at MD Anderson, translational science in pathology, pancreatic cancer, and any other type of cancer. You know all that he is a professor in pathology, and in my department also, translational molecular pathology. He's a scientific director of the Zayed Center for Pancreatic Cancer, but not for this Anirban prize. It prize for the science that he is doing. He is publishing wonderful science with immediate impact for the patients and for the very collaborative way to be. You have only to send an email to Anirban, look, I have this idea, can you help me? And in the same minute, you get back the answer, positive answer, and everything what he can do to help us to move forward the science at MD Anderson. With this said, Anirban, it's our pleasure to be here with you. We are very curious for your presentation, and good luck with everything.
Thank you, George. Thank you so much for the invitation and the checks in the mail for that wonderful introduction. So I also want to thank Emily and Liz for inviting me to speak today. I think, and of course for the Enjoy Science swag that I'm wearing, it looks really great. I hear that they're going to have this available at some point for anyone who's interested. T-shirts are fantastic. So I'm going to speak a little bit today about emerging molecular targets in pancreas cancer. This is the lovely building we are in. The pancreas cancer is on the 3rd floor of this really beautiful building. We're across the corridor from the molecular diagnostics lab. This is also where the Institute for Personalized Cancer Therapy, or IPCT, is housed. And so just a really great collection of scientists and physicians here. So moving forward, when we talk about tumors of the pancreas, it might come as a surprise to some, but not to everyone necessarily. But there are a lot of different types of cancers in the pancreas. The one that I'm going to speak about most today, and which is the focus of a lot of research in my lab and around the country, is ductal adenocarcinoma of the pancreas. But really, there are many other tumor types, including neuroendocrine tumors, acinar cell carcinomas, even tumors that occur in children, like pancreatoblastomas, etc. But for the most part, my talk today will actually focus on adenocarcinomas. So just a little bit of pathology. After all, as George said, I am in the Department of Pathology, and so it's obligatory that I show some H&E slides. So this is really what we are looking at when we are looking at tumors of the pancreas. You can see here this intense stromal reaction, or what we call stromal desmoplasia. And embedded in that are these neoplastic glands, and that are trying to recapitulate normal glands, but not doing a very good job at it. Here's a nerve, and pancreatic cancers have a great propensity to invade nerves. One of the reasons why so many patients present with intractable mid-back pain is because of that. And I'll show a picture coming up soon of what that looks like. This is an example of lymphatic invasion. This is a lymphatic vessel, and you can see tumor cells sitting inside it. And this is one of the reasons why so many patients, unfortunately, present with metastatic disease that is either regional or distant metastatic. This is an example of vascular invasion. The portal vessels are right next to the pancreas, and it's not uncommon to find the tumor cells invading the portal circulation and going to the liver. And that's one of the reasons why the liver is the most common site of distant metastases from pancreatic cancer. And then this is the neural invasion I was talking about. You can see this is a nerve ending here, and you can see the tumor cells have invaded into the intraneural space. Sometimes they're sort of around it or perineural, but in this case, they're actually intraneural. And this is also a way of egress for these cancer cells. So we talk about lymphatic invasion, hematogenous dissemination to the blood vessels, but you can also have neural invasion and dissemination by that pathway. So in the last year, we had a lot of well-known names that unfortunately succumbed to this disease: Justice Ginsburg, Representative Lewis, Alex Trebek. But I also wanted to show this series of pictures that I think really highlights the fact. This is Patrick Swayze, the actor who passed away from this disease, and you can really see that pancreatic cancer, even though we think of this as a tumor in the pancreas, is really a systemic disease, and there is a profound muscle wasting or cachexia that happens in individuals, especially with terminal disease, unfortunately. And so I'm not going to talk about this particular aspect of it, but for those of you who are in this field or who take care of patients, are obviously well aware of this phenomenon and why supportive care for patients is so important over and above what is done in terms of chemotherapy or radiation therapy. Besides treating the tumor, you also need to provide the systemic support to improve survival of our patients. So there is a rising burden of pancreatic cancer deaths, as many of you might know. Pancreatic cancer is the third most common cause of cancer-related deaths, even though its incidence accounts for less than 10%. But it is the third most common cause of cancer-related deaths after lung cancer and colorectal cancer. And it's actually forecast to become the second most common cause of cancer-related deaths in the next decade or so. Even the state of Texas here, you can see the numbers of cases and deaths are rising each year. And part of this is multifactorial. We have an aging population in this country. The median age of pancreatic cancer is about 69 or 70 years. And so as the population ages, we're seeing more individuals get diagnosed with this disease. There's also better classification that we have now. When somebody gets a tumor that's metastatic, it's not, in the earlier decades, it would be called carcinoma of unknown primary, but now with better molecular classification, we're calling these tumors pancreatic adenocarcinomas because we know where they're coming from. But there's also a real increase. It's not just a relative increase, there's also a real increase. And part of that is driven by the fact that other health conditions like diabetes and obesity do increase the risk for pancreatic cancer. Unfortunately, especially in the Western world, there is an epidemic of what we call the diabesity epidemic, and that has certainly contributed to the rise of this cancer. So in 2008, Dr. Vogelstein and colleagues at Johns Hopkins sequenced the first series of the pancreatic cancer genome, so to speak. This was 23 cell lines and patient-derived xenografts published in Science. And always comes as a surprise. This was 2008, which is not that long ago. We're not talking about the 1980s here. But this was all done by PCRs. This was before the next generation sequencing revolution, so to speak. And it's hard to think now for so many grad students and postdocs who do sequencing overnight for their projects, just how intense and labor intense and time consuming this process was, even just in 2008. Since then, obviously, there's been a lot of progress in terms of sequencing efforts in this disease. We have large data sets, including from the Cancer Genome Atlas or TCGA and the ICGC, which is the International Cancer Genome Consortium. All of these are publicly available and have really contributed to our understanding of the pancreatic cancer genomic landscapes. And so the big enchilada, if you so will, in this disease is obviously KRAS. KRAS is the biggest player in pancreas cancer. More than 90% of pancreatic cancers have a RAS mutation. And wonderful seminal studies done by Dave Tuvison, Sunil Hingorani, Andy Aguirre, Ron DePino, and others showed now almost 20 years ago that when you misexpress a mutant KRAS in the pancreas of mice, these mice develop precursor lesions just like you see in the humans. And a subset of these mice go on to develop primary adenocarcinomas and even metastases. And then the influence of cooperating mutations on this mutant RAS background is to alter the penetrance of the disease and alter its latency. But at the end of the day, you need this RAS mutation to develop this series of precursor lesions and invasive adenocarcinoma. And then other studies, including studies done by Hao-Chang Ying, now here at MD Anderson, and Ron, when they were at Boston, showed that if you turn RAS off in established tumors, these tumors tend to regress. And that's also been shown by investigators like Marina Pasquini-Magliano at Michigan, et cetera. Interestingly enough, now we know that these tumors don't really go away altogether over time. They come back through other resistance mechanisms that emerge. And in fact, we are seeing some evidence of that in the context of what's happening in the clinic as RAS targeting becomes a reality. Which brings me to the next slide, which is for the long time, RAS was considered the undruggable target de jour in cancer. RAS and MYC, et cetera, have always been in that group of oncogenes that are considered undruggable. But now we have really entered, so to speak, a really remarkable era of RAS targeting, direct RAS targeting, I should say, not indirect, but direct RAS targeting, thanks to the availability of these small molecule inhibitors of RAS. The one that's already in the clinic, and actually the first one that's been approved for non-small cell lung cancer, is the Amgen sotorasib drug, or what used to be called AMG 510. And then a whole bunch of other companies are coming up with their G12C assets, including Mirati and others. And then the most exciting one, which many of us in pancreas cancer field are waiting for, is obviously the G12D inhibitors, and to some extent the pan-RAS inhibitors as well. And the reason for that is, well, the G12C is the one that was first off the out of the gates because of just the chemistry of the cysteine residue. It only accounts for about 1.6% of pancreatic cancer mutations. And again, I would have to say that MD Anderson has been at the forefront of the G12C pancreas trials that have been done, or the patients that have been treated on these G12C basket trials. I think probably the investigators here, like David Hong and Shubham Pant, have probably the largest experience on the planet of treating G12C pancreas cancer patients. but it's still only 1.6%. And so we really are all eagerly awaiting the clinical release, so to speak, of the G12D inhibitors and the pan-RAS inhibitors, which are currently undergoing preclinical testing. And then there are also other agents, including inhibitors of SOS and SHP2 that we know are involved in adaptive resistance. So already, even before the first set of G12C inhibitors got approved or in clinical trials, preclinical studies have already shown that, that there are adaptive mechanisms that come into play, and these patients tend to progress on monotherapy alone. And so already combination trials are ongoing with some of these drugs targeting these adaptive resistance mechanisms. So I do want to pause for one moment and acknowledge MD Anderson's role in targeting RAS. And this is really work that came out of the lab of Raghu Kalluri here at MD Anderson, who's the chair of cancer biology. And this was a paper in Nature published about 5 years ago now, and was funded through the Moonshot at MD Anderson, where they used, this was a preclinical study that used MSC, mesenchymal stem cell derived exosomes, containing siRNA against KRAS, and they were able to show quite effectively that genetic targeting of RAS, remember this is not small molecules like I was talking in the last slide, genetic targeting of RAS was able to suppress tumor growth in multiple orthogonal models and enhance survival. Then E.J. Chapal and colleagues here in the stem cell transplantation department were able to implement a pipeline for GMP scale production of these MSC derived exosomes and containing siRNA against KRAS. So this was published the next year in JCI Insight. And now, after having gone through this entire process, we're very proud of the fact that the team that's led now by Shubham Pant and EJ has initiated a clinical trial of a phase 1 trial of treating patients with advanced pancreatic cancer using these MSC-derived exosomes targeting mutant KRAS. So this is, to the best of our knowledge, the only trial right now that is using an exosome-based genetic targeting of RAS, and it's against G12D. So we're not constrained by the G12C proportions. And again, this is an entirely indigenous effort led by MD Anderson scientists and physicians, and just something that we are very proud of. So now, obviously, there's a lot of interest in molecular profiling of patients, and certainly in the case of lung cancer and many other solid tumors, this is considered sort of the first pass that is done in any patient, and then they're kind of stratified based on the driver mutations that are present into different therapeutic subsets. But for a long time, there has been a reluctance in profiling patients with pancreatic cancer with the thought that, well, it's all KRAS anyway, and nothing else is actionable. So we often ask the question, is there a rationale for molecular profiling in the clinical care of pancreatic cancer patients? And I would have to say that in the last just even a couple of years, really the field has come around to really buying into this idea more convincingly than it ever had. So some of this work was based out of a study that was published from Pancreatic Cancer Action Network, or PanCAN, in collaboration with Michael Pishwain at Johns Hopkins and many other oncologists, where they looked at a series of pancreatic cancers in the community setting. This is not academic cancer centers. And when they sequenced these cases, they found about 27% of cases had highly actionable mutations, which is a little high. I think this number overestimates the actionability, but it certainly is there. A more plausible number was actually published by Arthur Singhi and colleagues using Foundation Medicine data, where they looked at 3,600 patients. And now I think this is being expanded. There's a new study coming now with almost 25,000 plus patients in that series. But in this initial study, they found about 17% actionable mutations. And you can see that on the left. And what is very interesting is, in particular, patients who did not have RAS mutations. Remember, RAS mutations are about 92%. So there's an 8 or 10% of patients who don't have RAS mutations. In that RAS wild type subset in particular, there are a lot of actionable drivers that are present. And so for that subset, for sure, if you look for RAS mutation in patient's tumor and you don't find it, definitely make sure you chase down that tumor for these alternative drivers. Sometimes a DNA-based assay will not be able to find it because many of these tend to be fusions, expressed fusions. And so you might want to actually do an RNA-based fusion assay, and these are also available in many molecular diagnostics labs. So the summary of this is that, yes, there are actionable mutations beyond RAS. And of course, once RAS inhibitors are in the clinic full time, this is going to blow up. You might just have to do RAS mutation testing alone. And if that's not present, you'd have to look for other drivers. But even before that, there is very good evidence to suggest that you should definitely look for these actionable mutations that are present in a subset of pancreatic cancers. And so one of the first targeted mutations for which a drug was approved for pancreas cancer was BRCA mutations. This was germline BRCA1, BRCA2. And just before the pandemic hit, actually in December 2019, was when the FDA approved olaparib, which is a first-in-class PARP inhibitors for germline mutant pancreatic cancer. And now a lot of subsequent studies are ongoing, expanding this initial indication to other genes in the homologous recombination repair pathway beyond BRCA1, BRCA2. So here, for example, this is the only time in my life I'm ever going to be quoted in People magazine, so I always show this. So this is actually a gentleman who had one of these homologous recombination repair family genes called PALB2, which is also mutated in a small number of patients, just like BRCA1, BRCA2. It's in the same functional pathway and benefits from PARP inhibitor therapy. And so this is another great example. If you don't look, you won't find. And if you don't find, you won't treat. So look. There are other such alternative drivers, as I mentioned, especially in the RAS wild type family of tumors, that 8 to 10% of cases. So here, for example, is and this is not the only one. There have been other series like this that have been published. These are patients who all had ALK fusions. And these fusions are very similar to the ones you find in lung cancer. And we know these are not metastatic lung cancers to the pancreas, so true pancreas primaries with ALK fusions. They tend to be especially enriched in younger individuals, so those who have pancreatic cancer under 50. Often you will find these fusion drivers in their tumors. And so all of these patients in this particular series were under 50. They were all RAS wild type. And they responded. They responded to the ALK inhibitors that you are approved to use in lung cancers that were used off-label here. And so again, these are therapies that might benefit these subsets, minor as they are. But for them, this drug is what's going to enhance survival. Another small subset, 1% or so, are TERK fusions. And again, while RNA-based assays are probably ideal, you can even look for TERK fusions using immunohistochemistry. It's a really nice study from Eileen O'Reilly and at Sloan Kettering, and Jackie Heckman, who was at that time at Sloan Kettering, showing detection of these TERK fusions using immunohistochemistry. So based on all of this data, from PanCan's Know Your Tumor program, from the Foundation Medicine data, from examples like this. Eventually, last year in 2020, ASCO finally updated its metastatic pancreatic cancer guideline, basically saying that everyone should get germline and somatic testing, everyone with advanced pancreatic cancer. And the reason for that are these subsets that I just discussed, such as the fusions, the BRCA mutations, for which now there is an approved therapy, and of course, microsatellite instability, for which there has been a tumor agnostic immune checkpoint inhibitor therapy approved for a while now. And so don't neglect it thinking, oh, it's all going to be RAS and nothing else. And of course, once RAS inhibitors are here big time, that's all you might have to look for initially, and then reflex to a more extensive testing if there's no RAS present in those tumors. So one thing I wanted to talk about, just again from a pathology perspective, is all of this is fine and dandy, but unfortunately in our world, we are often left with very minute specimens just because of the nature of the tumor. Most patients present with metastatic disease. And so all you get are these IR-guided percutaneous biopsies or endoscopic ultrasounds. But the good news is that you can do a lot with less now. And so we have published earlier this year how you can get whole exome sequencing data, including copy number data from primary and metastatic US FNAs. And again, this is published, so you can look at the paper. And the quality of the data is absolutely comparable to what you find with resection samples. And again, this is whole exome. This is not even targeted sequencing. We do whole exome because we also get data in terms of the structural defects that are present that might tell you that there is a DNA repair defect present, even in the absence of an obvious BRCA mutation. And those patients will benefit from things like platinum therapy or PARP inhibitors. So this helps give that added layer of information. And it's not just DNA. Obviously, there's more to pancreas cancer than DNA. And so we can also do RNA and not just RNA-seq, but single-cell RNA sequencing from these tiny primary and metastatic core biopsies. Again, this is published a month or so ago, so I'm not going to go in detail, but essentially we are able to get highly reproducible data with all of the compartments represented, including the immune cell compartment, stromal compartment, epithelial compartment, map the heterogeneity of each compartment. And again, all of this data is published, so I'm just referencing this paper. One of the things that really helps us with this level of resolution into single cell level is that you can then use algorithms to identify potential ligand receptor interactions in the data set. And why that's important, this is important, especially in the setting of patients who are on treatment and you're trying to look for adaptive mechanisms. So for example, you might be able to find that there are certain pathways that are particularly relevant in the post-treatment biopsy that might then form the basis for subsequent lines of therapy. So many of these are known pathways that we know exist and are important in terms of the immune suppressive microenvironment, IL-1, IL-1RAP, or the CD40 pathway, or the recently described, there was actually a Cancer Cell paper on this just last month on CD155 TIGIT. So we see all of these signals come up pretty strongly in the primary and metastatic single-cell RNA-seq data from patients, again, suggesting that these are signals that you can look not only at baseline, but also in patients on treatment, and therefore identify potential adaptive resistance mechanisms. So this is the conclusion of the first part of my talk, which is a molecular profiling, including whole exome sequencing and single-cell transcriptomics, can be applied to limited real-world samples that we get from most of our patients. We don't typically get those large resections that many other tumor types have. And second, these single cell approaches can provide insights into the heterogeneity of pancreatic cancer, and most importantly, allow us to interrogate adaptive resistance mechanisms in patients on therapy. So I want to switch gear now and talk a little bit about one story, because again, because of time, it does not, I can't talk about all of the mouse models we are working on, but we do have several. We have done a lot of work in terms of annotating, functional annotation of driver mutations in pancreas cancer. That's one thing that our lab has done over many years, which is there's RAS, and then there are all of these cooperating mutations. And so we have systematically built these cooperating mutations into the RAS background, whether it's ARID1, KDM6A, MLL3, BRCA, GNAS. So these are all sort of these cooperating mutation studies that allow us to really understand in the autochthonous setting what is going on in terms of driving these tumors. And I'll tell you that having done this now, there's always a surprise, because autochthonous models often end up giving you surprising insights that are not often gleaned necessarily from cell line models or even organoid models, as good as they may be. And so there is I would say, a huge upside. These are labor intensive, time consuming, and expensive, but the insight that you can get as to what is exactly happening in terms of driving tumorigenesis from an autochthonous model really is hard to recapitulate. And so we have spent a lot of time and effort in making these cooperating mutation models for that reason. So again, going back to the TCGA for a second, these are all of the models that we have made in our lab. Some are published, some are not yet published. But the one I want to talk about today, which is not yet published, is it's going through the wonderful peer review process that we all deal with. But there's a bioarchive that you're welcome to refer to if you want. Is an RNF43 mutation. So just very quickly, RNF43 is mutated in about 7 to 8% of pancreatic cancers. And so as you can see in the TCGA ordering list, it's actually right after the big four: KRAS, P53, P16, and SMAD4. So it's not an uncommon mutation in pancreatic cancer. And so what is RNF43? It's a E3 ubiquitin ligase, and it is known to degrade a Wnt receptor, which is frizzled on the surface and therefore act as a negative Wnt signal regulator. It also has a putative role in inactivating the downstream TCF4 itself. But I think the main function that has really been well established is actually at the surface level in terms of degrading Wnt receptors. And great pioneering studies from Hans Clevers and others have shown that RNF mutation results in sensitivity to Wnt inhibitors, a porcupine inhibitor in colon cancer preclinical models. So one thing that kind of surprised us when we looked at RNF43, and we knew that it's mutated in the 7 to 8% of pancreas cancer. So we start looking at what does it do in terms of outcomes. And the TCGA data is on the left, and these are small numbers, so I don't want you to necessarily say, oh, this is a large series, but it still looked surprising to us that tumors that had RNF43 mutations tended to have a better prognosis. And then what really bolstered our confidence was data from David Chang, Stephen Dreyer, and others at the ICGC, where they showed that when you look at recurrence patterns, patients who have undergone surgery, and then unfortunately, the vast majority will recur in pancreatic cancer. The only mutation that is present at a higher frequency in the no recurrence subset is RNF43. So if you have an RNF43 mutation, you tend to not recur in a significant proportion. And so that got us thinking, maybe there's something to this RNF43 story in pancreas cancer that's interesting. In the meantime, while all of our mouse model work was going on, a couple of papers came out that showed that very similar to what was initially described in the colon cancer model, when you have RNF43 mutations, you have Wnt dependency. This was all work done in cell lines, by the way. So these are established cell lines, ATCC lines. And essentially what they showed was when you treat these cell lines with Wnt inhibitors, a porcupine inhibitor from Novartis, LGK974, these cell lines, you inhibited the growth of these cell lines, showing that Wnt was hyperactivated. When you lose RNF43, you upregulate this receptor, and therefore you upregulate Wnt. So based on this, actually, Novartis did a clinical trial right here at MD Anderson. Philip Janku, who used to be here till recently, was the PI of that trial. And unfortunately, as often happens in the preclinical to clinical translation, it didn't pan out. There was in fact no response in patients. This was a basket trial for RNF43 mutations. And so everyone in the trial had an RNF43 mutation, and including pancreatic cancer patients, and no one had any response. And there was only one partial response in an appendiceal adenocarcinoma. So again, this sent us back to the drawing board, saying, well, what happened here? You had all this data in colon cancer preclinical models showing that RNF43 loss upregulates Wnt signaling, and these tumors are sensitive to porcupine inhibitors. You had these two preclinical data, one in using a small molecule against Wnt, and the second one using antibodies against the frizzled receptor itself. that showed in cell line models that you had Wnt sensitivity. So what exactly happened? So that's why we ended up, again, falling back on our autochthonous models. And as I said, this is in revision gastroenterology, but the preprint is online and you're welcome to look at it. So what we found is when you lose RNF43 in the mutant KRAS background, you get cystic lesions, and then you get adenocarcinomas. And you can see that the adenocarcinomas arise even in the setting of haploinsufficiency, which is interesting and a whole different story we are pursuing. So we then took these tumors from different time points, early, intermediate, and moribund, and we did single cell RNA sequencing to figure out the composition of the tumor microenvironment and the immune infiltrates. And immediately it hit us that there's something going on that is very strange. It's not something we're used to see. And we've done a lot of single cell sequencing in different mouse models of pancreas cancer and human patients, as I showed before. The first thing we realized is these late stage tumors, they had way too many B cells and T cells. You're not used to seeing this in most pancreas cancers. You don't have so many B cells or T cells in pancreas cancers, and there was like bucket loads of it. Vice versa, and that's the green and the red that you see here, the green and the red. Vice versa, there seem to be fewer myeloid cells, which is again something that dominates the immune landscape of the usual pancreatic cancers. So we went back. Well, first thing we did was we then went and looked at the epithelial compartment only, and that's what single cell analysis allows you to do. You can partition by different compartments. And we saw that the Wnt signaling was really not important. I mean, it was there in the very early stages as one of the enriched pathways, but by the time these tumors had established themselves, Wnt was gone. And so that could very well explain why, maybe it's something that's important in early stage disease. Maybe it's important for interception in precursor lesions, but by the time you have established tumors, Wnt stops being important. And that probably explains why the phase 1 trial did not show any activity at all. So, then we, as I said, we started sort of deconvoluting the different other cell types that are present in the immune microenvironment. And one thing we realized was that the macrophages that are typically seen in your KRAS P53 models, they are significantly depleted. Instead, you have these inflammatory macrophages, arginase positive, that are responsible for this profound immune suppression that you see in pancreatic cancers. They're really suppressed over time. Instead, you have a lot of the monocytes and other cell types. So again, this is what the typical mouse model, the KRAS P53 model, looks like. This is from Dave Tuvesen's lab. This is data from Rolf Brecken. And you can see this big orange blob in the middle. That those are all macrophages, and we don't see that with the RNF43 mutation. So again, divergence in immune microenvironment of KPC versus KRC. The macrophages in red from the KPC model, they're not present, barely present in the KRC model. Instead, you have a lot of B lymphocytes and T lymphocytes and plasma cells. There's hardly any plasma cells in the KPC model, the KRAS P53 mutant model, but you see a lot of plasma cells. So again, the immune contexture was completely different with the RNF43 mutation versus P53. Let me talk a little bit about the B cells here. So again, this is something we really don't find in either human or murine pancreatic cancers. B cells, there are immune suppressive B cells that have been reported by Yulia Paleva-Gupta and others in the context of pancreatic cancers, but the antibody producing, pro immune B cells, if you so will, or anti-tumor immune cells, if you so will. There's hardly any report of that in adenocarcinomas, but we see a lot of that in the KRAS model. The other thing, again, coming back to the T cells, again, there is good amount of T cell maturation. But what is interesting is that there's also a lot of checkpoints. So there's a lot of T cells that you find, but there's also a lot of checkpoints that are expressed on these T cells, including CTLA4 and PD-1. So seeing that CTLA4 expression, and this is going back for a second here, this reminded us a little bit of the melanoma type microenvironment, right? You have T cells that are present, but those T cells have a lot of checkpoints on them, so they're not able to do their work. Now, if you give these mice or give these tumors and checkpoint inhibitor, you can basically activate the T cells and get immune rejection. And so we said, well, let's see if that really happens. And so we did this preclinical study where we treated these mice with anti-CTLA4, and this is mice that are KRAS RNF43 mutant. This is monotherapy with anti-CTLA4. And as many of you know who work with preclinical models of adenocarcinoma, the anti-CTLA4 monotherapy has no effect at all, literally no effect in the traditional models. But we were surprised to find that there was actually a pretty robust effect with just anti-CTLA4 monotherapy. So again, this is to summarize the findings of this RNF43 mutation versus the traditional P53 mutation mutant mouse model, which is compared to the KRAS P53 model, there's a lot less myeloid cells, there's more T cells, much more B cells, and plasma cells. Again, I can't emphasize this enough. For somebody who looks at pancreatic cancer, human pancreatic cancer samples, there's not a lot of plasma cells at all. And so we find a ton of plasma cells in here. So why is this happening? Why is there this difference in this immune contexture? And, I'm just cutting short a long story of looking for things. But the bottom line is it boils down to the complete absence of this pivotal cytokine that has been reported by many investigators looking at the microenvironment of pancreatic cancer, which is CXCL5. The lines that from the KRAS P53 model are on the top, and the KRAS RNF43 model are at the bottom. And you can see there's complete absence of any CXCL5 in these RNF43 mutant tumors. And not surprisingly, when you re-express CXCL5, in the RNF43 mice, you end up getting the tumors to grow and kind of start behaving like a KRAS P53 model. These tumors are larger. They start having myeloid infiltrates, less T cells. So basically, you've converted a KRAS RNF43 mouse into a KRAS P53 model simply by re-expressing CXCL5. And so why is there no CXCL5? It's an epigenetic regulation that happens. We can see that there is no transcriptional expression. Of course, there's no protein expression, no secretion, there's no transcriptional expression. And there's also an epigenetic suppression of CXCL5 expression that happens. And so we also went back and looked at CXCL5 expression in pancreatic cancer samples that are RNF43 mutant versus not. And you can see that they actually have significantly lower CXCL5 expression as well. So what is suppressing the CXCL5 expression? Again, this is a long story. I'm cutting right to the chase here for time. We have to finish soon. Essentially, there is a high expression of HDAC8. So loss of RNF43 in pancreatic cancer stabilizes this particular HDAC, which is HDAC8, not the other HDACs. And that leads to transcriptional suppression of CXCL5. And so when you inhibit HDAC with either a pan HDAC inhibitor like panabinostat or a more specific HDAC8 inhibitor, which is this pharmacyclics compound. It's not in the clinic, it's a preclinical compound, PCI 34051. Both of them, when you suppress HDAC activity, and particularly HDAC8 activity, you can re-express CXCL5. So again, one of the cautionary notes I would say out of all of this is that people have used HDAC inhibitors in pancreatic cancer, and they have not been very successful, to be honest with you. And I'm concerned that part of that could be that we might actually be doing harm in altering the immune microenvironment and making it more sort of immune suppressive by upregulating CXCL5, even in tumors where CXCL5 may not be expressed or may not be expressed at high enough levels, but we could be essentially impacting a regulator of CXCL5 expression here and with these HDAC inhibitors and making these tumors worse. So again, what we believe is happening here, and again, this was not something that was obvious with cell line data, would not have been obvious with organoids, and again, really underscores the importance of spending the time and doing these autochthonous models to understand the differences between different genetic backgrounds. So when you have KRAS P53 models, you have low HDAC8 levels, increased CXCL5, you have decreased lymphocyte infiltration. These tumors are refractory to IO. Vice versa, with RNF43 mutations, you have high HDAC8, low CXCL5, you have increased TME. So it kind of looks like a melanoma-ish TME, as I said before. And so maybe these tumors, instead of trying porcupine inhibitors that were tried and did not work, maybe these are the subset where we can really go all in with IO, select those patients and see if there is even a marginal benefit or we can enhance the benefit with IO combinations. So this is a clinical trial that's ready to be written. And if there's an oncologist who would love to do this trial, we would love to work with you. So the conclusions on our second part is autochthonous models. I can't stress this enough, provide mechanistic insights, international history and the microenvironmental architecture. Long sentence, but the bottom line is they tell you things that a cell line or organoid won't do. And we think that RNF43 mutant PDAC might be a subset that's amenable to immunotherapy because the paucity of CXCL5 and these immunosuppressive myeloid populations might be creating a microenvironment that is more amenable to immune-based therapies. So that was the last slide. And I want to thank everybody in my lab, including a lot of the alumni. In fact, I just realized this morning as I was looking at the slide, there's a lot more alumni. And so it's not that our lab is empty. We are a big lab, and we are always looking for wonderful, talented postdocs, grad students, whoever wants to come and join our endeavors. But really, there's been some really great people who have done a lot of the work that I presented today. I want to thank all of them. I want to obviously thank our clinical collaborators, without whom a lot of the human tissue work I presented would not have been possible. And then the MD Anderson Moonshot, the Khalifa bin Zayed Foundation, and the NIH, which funds a substantial amount of my research. Thank you very much.
Thank you very much, Anirban. This is a wonderful talk. And in fact, you will see a lot of people already start sending chats about your fascinating presentation. Thank you very much. Let's start with a question, the most interesting part of the story. So, first question, did CXCL5 increase may increase the B cell counts in the tumors?
CXCL5 may increase. No, I don't know. I don't think so. I think that there's lack of CXCL5 in the tumors, and there's no myeloid cells as a result of it. And I think there is data certainly from David DiNardo and others that have shown that just even getting rid of myeloid cells, it's almost like creating a space, might be enough to get T and B cells into a tumor. Here, you're basically naturally creating a myeloid depletion environment. And so that gap may be being filled by T and B cells. We did not find a specific cytokine that's bringing the B cells in, if that's what the question is. And I have to say that the story is not complete. There's a lot of other things going on, but we didn't see anything specifically bringing the B cells into the microenvironment.
Thank you. Now, another quite general question. So as pancreatic cancer, it's a deadly type of cancer, and we have all these new technologies. What do you think has to be done to get through the truly early diagnosis to save the lives?
Wow, that's my 1 hour talk. I didn't give that talk. So I think first of all is to understand who is at risk. We can't go about just screening everyone for pancreas cancer, because I think there's a lot of chances for doing harm and false overdiagnosis, etc. And so you have to first define who is at risk. And we have very good idea of who's at risk. And there's some really wonderful work done here by Florencia McAllister and others in the high-risk clinic, showing that there are families that have genetic mutations. About 10% of pancreatic cancers arise in patients who have a germline mutation, including the BRCA mutations I talked about in the treatment context. And so family members of patients who have BRCA mutations or PALB2 or other of these germline mutations, they should undergo testing to see if they're at risk. If they carry a mutation themselves, they should potentially talk to a genetic counselor or a physician about getting screened. And Florencia is actually the director of MD Anderson's high-risk clinic. She and Michael Kim run a wonderful operation here, looking at patients with these high-risk characteristics. The other high-risk feature are patients who have pancreatic cysts, and Michael Kim is actually the director of the cyst clinic. So they always sort of work together. So there are these defined high-risk populations where you can potentially do your biomarker studies. And you do that a lot, George. If you're interested in a link RNA panel or microRNA panel, don't go around doing this in the general population. You need to focus this initially, at least in the high risk, and say, is it working in the high risk? And if it's working there, then you have to slowly build it to the sort of the average risk population.
Yes, thank you very much. Very interesting point. Yejin Gay is telling you wonderful talk, and she is asking generally, how do you think to convert KPC-like cold tumors into KRC-like more sensitive to immunotherapy tumors? Has this to be done through wing signaling high states? So how do you think to convert cold in hot tumors?
Yeah, I think that's a great question, and I think that's kind of the whole holy grail, if you so will, of making immunotherapy work in pancreatic cancer. I think there are multiple sort of avenues that can be taken. The biggest one, I think, is again, you have to go after the myeloid population. I think RNF43 mutations is a wonderful natural experiment because it tells us what's possible in pancreas cancer when you don't have a lot of myeloid cells. But I think if you don't have that opportunity, then you have to go after the myeloid cells. And certainly that's being done in the clinic right now through various strategies, targeting different CXCR receptors, et cetera. The other thing is there are other, obviously, the T cell activation is an important part of all of this. You can't just do one thing and not do the other. I mean, even here in RNF43, there are T cells, but they have a ton of checkpoints on them. So that part has to happen, but that has to happen in conjunction with some of the myeloid depletion or reprogramming strategies as well that need to be done. And this is why the only signals that have come, and again, immunotherapy by no means is a success in pancreatic cancer, but the only signals that have even come conceivably have been in those regimens that have used a combination of more than one approach that goes after the myeloid compartment and the T cell activation strategy together, often with chemotherapy as well.
Great. Another question. Have you checked the tumor microenvironment of SNF42 mutated tumors?
RNF43 mutant tumors, you mean? Yes.
The tumor microenvironment.
Yeah, so they have reduced. That was the data I showed from LCM data that showed that there is reduced CXCL5. What we haven't done, and I need to do that next, is to confirm the increased B and T cells in the same patient. So the CXCL5 is just an expression data. So at least that angle looks to be panning out. But is it also accompanied by the other parts that we see is something that we need to look at. Now, again, as I said, we started this project because of the correlative TCGA and ICGC data that showed a better response, better outcome in these patients. And so there's clearly something going on in the setting of RNF43 mutations that's causing these individuals to have either overall better outcome or no recurrence. And so clearly we have to now figure out whether the mouse model data is panning out in terms of the immune contexture as well. But the CXCL5 is true. That we have looked at already. That was in the talk.
Thank you very much. So there are a lot of questions. I move forward. How did you zero in on this particular HDAC8 and if this HDAC8 it's a direct target for RNF43 mediated ubiquitination.
Yeah, so that's a great question. So that's exact. So HDAC8 is not a direct target of RNF43 ubiquitination. I think Sharon, you asked that question, if I remember correctly, and obviously that was the first thing we looked at, and the answer to that is no. And so we are trying to figure out why is HDAC8 up, and what is that arrow between loss of RNF43 and HDAC8 being up, and why are the other HDACs not up? So that is obviously one of the big unanswered questions of the story, which is the arrow. We have some ideas that it is mediated by a stress response. There's a whole mitochondrial stress response story here that I haven't even talked about. So that I didn't have the time to talk about here. But we believe that loss of RNF43 also causes an entire mitochondria phenotype that's possibly being reflected in the HDAC8 going up, that's then causing the CXCL5 to go down. Okay, so that's being chased in the lab as we speak. But it's not a direct target.
Another question. Is there a hypothesis as to why the myeloid cells are lower in the RNF43 mutation? I'm wondering if there is a mechanism that can be used to target the myeloid cells in RNF43 wild type mice.
Yeah, so that's the CXCL5. And so in fact, targeting that pathway or CXCR inhibitors, that's exactly what's being done. Because I mean, again, I can't repeat this enough times. RNF43 is a natural experiment. The mutation is a natural experiment that tells us what can happen when you don't have those myeloid cells. So what people are doing in the clinic in the other tumors, which is the vast majority of pancreas cancers, is targeting those myeloid cells through various small molecule or antibody-based approaches in order to sort of mimic what we are seeing here. One could be a very facile way to do this potentially would be the other thing that I didn't mention is that there are other sort of inhibitors that we can use here, and we haven't tested those obviously in the context of altering the microenvironment in KPC models to see, if you can mimic what's happening in the KRC model, for example.
Wonderful. Other question. I keep going quick. Does mutation in RNF43 equals increased nuclear beta-catenin and the TCF7L2 activities? This is because the inhibitors works way upstream.
So this is the thing, and I don't know if this is, and again, I think this is why organ-specific context matters. What is true in colon or in lung may not necessarily pan out in pancreas. We don't see this Wnt angle in pancreas in our autochthonous model. There is a whiff of Wnt activity early on, and then that disappears, it's gone. And we don't see an increase in beta-catenin, we don't see an increase in any of the Wnt pathways. So I'm not sure if the Wnt effect that we see in colon cancer that is profound, and that was the basis for a lot of the work with RNF43, is necessarily true in pancreas. And part of it could simply be that there is, RAS is playing another role here, and that could be modifying what's happening. But it's not a dominant player at all in our pancreas model.
Great. Other question. Can you comment on inflammatory macrophages decrease upon RNF43 mutation? What are the markers for inflammatory macrophages?
Yeah, so arginase 1 is the most important one. And I think I had a figure in my talk showing there's a real serious reduction in the arginase 1 levels in the tumors, and also the macrophages that are reduced. And there are other inflammatory macrophage markers that we have used. I don't remember the exact sort of anchor panels, but on the single cell RNA data, you can see a very clear reduction. The other thing that happens besides inflammatory macrophages is granulocytic MDSCs. The granulocytic MDSCs are very, very quiescent in contrast to KPC mice, where you see them lighting up, really like the activated granulocytic MDSCs. In the KRC models, they're very quiescent, they're kind of quietly sitting there, again, showing this sort of immune suppressive microenvironment is really not happening in these mice.
Wonderful. Other question. So a great talk. When you saw expansion of T cells in your model, was there expansion of a specific T cell subtype, naive memory, and so on? And also combined question, does high density of tumor infiltrating B lymphocytes means of prolonged overall survival?
Yeah, I mean, so the answer to the first one is that we see an increase in actually in regulatory T cells and effector T cells, both. And again, all of that is in the preprint. You can go and check the data. If you need any of the single cell RNA sequencing data set from us, we're happy to share it. Now it's, as I said, it's a preprint, it's public. So there's an increase in several T cell subsets that happens. And there is an increase in checkpoint expression on these T cells. In terms of the B cells, so the other thing I wanted to add is we did look for specific clonotypes on the T cells, and I don't believe we saw anything that was particularly clone restricted here. There was actually several different subclonal expansion that happened here. The second thing is the B cell question. What was the question with the B cells?
The question about B cell is if you see if the B cell density in the tumor infiltrating B cell lymphocytes and plasma cells signifies prolonged overall survival. So if the B cell and plasma infiltration has to do with survival.
It could be that there is an ADCC effect going on against the tumor. I mean, we always talk about T cells and T cell cytotoxicity, but ADCC is also an important mechanism of tumor cell death. And it's very possible that the antibodies that these mice are producing from these plasma cells are also having a collateral benefit in terms of the survival of these mice. So we haven't looked at this in patients, but all I know is, for example, when we look at the pancreatic cancer microenvironment, or we look at progression of, say, precursor lesions to high grade and cancer, one of the features that we find is a reduction in B cells and plasma cells. When you take low grade lesions to high grade lesions to cancer, one of the consistent features is the B cells go down and the plasma cells go down. So the fact that not only are they not going down, but there's so much of it in these models tells us that they're obviously doing something.
Thank you very much. There are very many other questions because of
reach out to me, email me.
Please contact directly Dr. Anirban Maitra. It's also a question, is the presentation going to be posted in the intranet? I think your presentation will be, so thank you very much. With this, Anirban, thank you. It was a wonderful presentation. In fact, very many questions. I want to thank to all of you who attended and to tell you, enjoy science. These shirts, please contact Liz Kent and Emily Lang, because of course you can order the shirts and you can tell to everybody how nice the talks are in Enjoy Science, and basically how beautiful it is to do science in MD Anderson and to have fun making discoveries. Thank you all. Thank you, Anirban, and good luck, and see you in 2 weeks.